hardest parts about reviewing for any exam is getting the vocabulary right. So in this video, I'm going to go through some of the most important vocabulary words for any biology end of course EOC exam that you should probably know before you take the test. I have other review videos too that are more content and skills focused, so go ahead and check out some of those other resources. I'll put the links in my description below. Let's get started with our key vocabulary. All right, starting off with prokaryotic. Now this is a type of cell. A prokaryote is a prokaryotic cell, and it's an organism that lacks a nucleus and also does not have membrane-bound organelles. So no mitochondria, no Golgi apparatus. Eukaryotic organisms are organisms that have DNA in their nucleus, and they also have membrane-bound organelles. So these are organisms like plants, animals, protists, you and me. Think you, eukaryotic, you and me. All right, moving back to our organic compounds. Now, organic compounds, sometimes called biological macromolecules, are these main essential molecules that are necessary for life. Things like nucleic acids, proteins, carbohydrates, lipids. So let's go through each one of these. Nucleic acids are made of nucleotides. These are things like DNA and RNA. Proteins are made of amino acids. These are proteins, including enzymes. And carbohydrates are going to be our sugars, like our monosaccharides, our polysaccharides, and our starches, usually our very large polysaccharides. Lipids can include things like our phospholipids, regular lipids that are made of fatty acids and glycerols, and steroids as well. Also, it's important to remember things about enzymes. Now, there's a lot of vocabulary associated with enzymes, which we'll get to in a minute, but enzymes are proteins, so they're part of that category of organic compounds or biological macromolecules. So let's talk about some words associated with enzymes. Enzymes, you always want to think proteins. Enzymes are specific to the molecules that they act on. They're also sometimes called biological catalysts, meaning they help to speed reactions up or get reactions going. What that actually means is that they lower the activation energy of a reaction. So a reaction that might occur, they're going to lower the activation energy for it to begin. Enzymes remain unchanged after the reaction, and so that means they are reusable, and they're going to usually work best in a specific environment. So you might hear the word the optimum pH for an enzyme, and that's going to be if you see a graph where the peak on that graph is where the highest enzymatic activity can be. In certain conditions, for example, if the environment gets too hot for an enzyme, it can unfold or denature. That's another word to describe how an enzyme three-dimensional shape will unfold, and then that active site can no longer fit with the substrate, and we will have the enzyme not being able to work. So let's talk a little bit more about enzyme vocabulary here. So we have usually an enzyme that acts on a specific substrate. That's the molecule that the enzyme works with. And then afterwards, after the enzyme helps that substrate undergo its reaction, we'll have the products. And it can be a variety of different things. Sometimes it's bringing two molecules together, creating two new things. But when it's together, sometimes that's called the enzyme substrate complex. And the place in which the enzyme is going to interact with that substrate is the active site. Now, if the molecule, the enzyme, denatures, unfolds, the active site isn't going to be the same shape anymore, so it won't be able to act with the substrate. All right, let's talk about the phospholipid bilayer. This sounds like a big scary word, but it is just what our cell membrane is made of. Now, there's other things involved in the cell membrane, like carbohydrates and embedded proteins, but the primary structure of our cell membrane is that phospholipid bilayer. And so this is just a cross section, or like a straight on view of what the side of a cell membrane would look like. Obviously, this is a three-dimensional structure that surrounds the entire membrane. But we're just like looking up close at the cell membrane here. It's selectively permeable, meaning it's going to let certain things in, certain things out, but not others. So that way it helps protect the cell, helps maintain homeostasis, that internal balance that's all organisms need to maintain in order to survive. And what we see here are these molecules on the outer layer of the cell and on the inner layer of the cell. We see that there is a concentration gradient here. What that means is the difference in concentrations, or kind of amounts per volume, of a substance between two areas. So substances are naturally going to move from higher concentrations to lower concentrations. So in this case, because of the concentration gradient, we're going to see that this molecule here is probably going to go down the concentration gradient into the cell if we're pretending that this is outside and this is inside the cell. All right, let's talk a little bit more about that cell transport. So naturally, molecules are going to move from a higher concentration to a lower concentration. That's just diffusion. And I like to think of it, imagine like high to low, if we're represented that by a slide, like the kid's at the top of the slide going down, naturally he's just going to go down the slide. Facilitated diffusion is the same movement. So molecules are going to go from a higher concentration to a lower concentration, but with the help of a transport protein. And so in that case, if we're using our slide metaphor here, we can imagine a kid going down with the help of a parent or a friend. That's facilitated diffusion. High to low, still no energy required, but we do have the help of a transport protein. Active transport, however, is when we are going to go up the concentration gradient. So we're going usually from a lower concentration to a higher concentration, or it's a larger molecule and it requires energy. And in the cell, that energy is ATP. Now, the way I like to remember this is that active transport requires energy, and it starts with A and T. And so naturally, you're going to think AT, okay, ATP, energy required for active transport. And you can also imagine it if we go back to our slide metaphor, like the kid is going up the slide, he's going from low concentration to high concentration, and that requires a little bit of energy. All right, here it is without slides, just molecules, and we can see in active transport, if we start over here, going from lower concentrations to higher concentrations across the membrane, we do need that energy molecule, ATP. Moving backwards with facilitated diffusion, obviously we're still going from higher concentrations to lower. If we go in this direction, does not require energy, but it does require this little protein channel here. And then simple diffusion, high to low, no energy required. There's one more special type of movement that I want to talk about really quick, and that's osmosis, and that's the movement of water across a selectively permeable membrane, and it's really just how diffusion works itself too. So water will go from higher concentrations of water to lower concentrations of water, and so sometimes that means that cells will swell or shrink depending on the behavior of other particles around them. Okay, moving on to the ecology side of things, a heterotroph is an organism that must consume other organisms for food. So troph means feeding, that word part means to feed. Hetero means different. So different feeding, you gotta eat different things to survive. So humans can obtain nutrients by consuming other organisms or the products of other organisms. We are heterotrophs. An autotroph is kind of the opposite. Auto means self, troph means feeding. So an organism that is able to form its own organic molecules for food. So an autotroph is kind of self-feeding, like plants. They make their own food, they make their own glucose. You might see something like this called an ecology pyramid or an ecological or energy pyramid. And here we are looking at different trophic levels, different feeding levels, and seeing that as we go up in trophic levels, usually there's going to be a loss of energy. At the bottom of our energy pyramid, we have producers. You've probably heard that term before. And then as we go up, we get into the heterotrophs or different types of consumers. So here we have primary consumers, secondary consumers, and tertiary consumers. Sometimes at the top of a pyramid, you'll see an apex predator. That just means the predator that feeds at the very top level in a particular ecosystem. Symbiotic relationships are a type of ecological relationship where two organisms are going to share a close interaction in some way. They can be positive, like mutualism. They can be negative, like parasitism, or one can be helped and one may not care at all, and that's commensalism. But usually these types of behaviors are taught and tested at the middle school level. So for a biology 9th grade exam, you might not need to spend too much time focusing on the different types of symbiosis. But just know that organisms are going to interact with each other in various ways, for example, in mutualism. All right, let's talk about cells for a little bit too. We're going to go through different parts of the cell, and I want you to try to think of what the cell part is before I tell you. So let's start here. A large fluid-filled sac that stores nutrients and helps maintain cell structure in plants especially, but it can exist in animal cells as well. Do you know what it is? That's the vacuole. Good. And so usually in a plant cell, we'll see one very large vacuole, but vacuoles can exist in animal cells as well. A membrane-bound sac that contains enzymes that break down waste products and damaged organelles. Think you know it? That's the lysosome. An organelle responsible for modifying and packaging proteins and other molecules for transport within the cell or for export from the cell. That is the Golgi apparatus. All right. This structure is a network of tubules and sacs that helps transport proteins and other molecules within the cell. That organelle is the endoplasmic reticulum, and we can have the smooth endoplasmic reticulum and the rough endoplasmic reticulum, or the RER. The RER has the ribosomes, so think R for ribosomes, R for rough in rough endoplasmic reticulum. All right, small structures that produce proteins, we were just talking about these. Those are the ribosomes. A rigid structure that surrounds the cell membrane of plants and some other organisms, that is the cell wall. Moving right along, the gel-like substance inside the cell membrane, that is The cytoplasm. Right, an organelle in plant cells that uses sunlight to produce food. We'll talk about this more in a minute. That's the chloroplast. All right, and the site of energy production within eukaryotic cells, mitochondria. All right, let's go back to DNA. So that genetic material, it's deoxyribonucleic acid. It's made of nucleotides. This is one nucleotide here. A nucleotide contains a phosphate, a sugar, and a base. That base is either going to be A, T, G, or C. And all living things contain DNA. Here's a bunch of vocab for you. So DNA is found in the nucleus of eukaryotic organisms or in the nucleoid, so that mass of genetic information, in prokaryotic organisms. Remember, prokaryotes do not have a nucleus. Adenine is always going to pair with thymine. Guanine is always going to pair with cytosine. Those are our bases in DNA. Protein synthesis, that's that whole process of going from DNA to RNA to protein. Synthesis means making. Protein synthesis is just how proteins are made, but there's two main steps and they sound very similar. We've got transcription and translation. Transcription is going from DNA to RNA. Translation is going from RNA to protein. And the way I like to remember this is that we're going from nucleic acid to nucleic acid here. So it's like we're just writing down a message from one person to another, same molecular language. But when we translate, like we go from English to French, we're changing molecular languages. And that's what happens when we go from RNA to protein. We're changing the macromolecule that this message is in. So it's not a message anymore, it's how the proteins are built. So translation is when we take the RNA message and it is translated, it is changed and put together into a protein. All right, so again, protein synthesis, remember, starts with DNA. The mRNA message leaves the nucleus, and then the protein is assembled at the ribosome. All right, amino acids, remember, that's how those proteins are made. So when we get to that point in translation at the ribosome, tRNAs are going to bring over amino acids and link them together through peptide bonds, that's another vocab word, to form these polypeptide chains. And the amino acids are then going to fold together based on their properties to create 3D shapes that are the proteins. All right, let's talk about some more genetic stuff. So this is a very bad representation of a chromosome. I don't like this drawing at all. Chromosomes are more like this, where we have all of the DNA wound together. Remember, the DNA in organisms is super, super long, but when the DNA is just in the cell by itself, sometimes it's more like this kind of spaghetti form and it's not condensed. It's called chromatin. And so it's kind of confusing because we have also chromosomes which can split apart, and those pieces, those duplicated sides, are called sister chromatids. Okay, so here we go. One whole chromosome, chromatin, just in the nucleus, and then sister chromatids. And at the middle, we have a centromere here. All right, but when a cell prepares for division or mitosis, often the DNA is going to condense into the chromosome so that it can prepare and get ready for cell division. So let's talk about all the stages of the cell cycle. So these are just the series of stages that the cell goes through from birth to division. And it's going to have multiple checkpoints and spots along the way to make sure everything is going correctly. If these checkpoints are not followed, we can have uncontrolled cell growth or uncontrolled division, which leads to things like cancer. So the cell cycle is regulated by specific proteins, but there's some phases you should be familiar with. G1 is our growth or gap 1 phase. S is when we have DNA replication, that's the synthesis phase. G2, another growth or gap phase. And then finally M phase, which stands for mitosis. And technically some people put cytokinesis as its own stage there as part of the cell cycle. That's when that cell actually physically splits apart. All right, so once again, DNA replication happens during S phase, and it's going to allow our daughter cells to have exact copies of the parental DNA when division happens. So we need to have that DNA replication occurring before cell division, or else if we just split the cell in two without replicating the DNA first, we would end up with half the DNA in the daughter cells, which is not what we want for regular old mitosis. Now, mitosis is that type of cell division where we take a diploid cell and it produces two identical diploid daughter cells. That's a lot of vocab right there. Okay, so this is going to be for growth or repair. Like if my skin cells are going to grow, they'll go through mitosis. If I have a cut, again, mitosis. Cancer is too much mitosis going on in the body. Mitosis is going to start with one cell. Remember, the cell has already duplicated its DNA, and then it's going to go through its own stages before it divides. So those stages are, you can remember PMAT. Another way I like to say is Prophase, metaphase, anaphase, and telophase. That's a lot of words. Let's see them together. Prophase is that preparation. Metaphase, the chromosomes are lining up in the middle of the cell. Anaphase, the sister chromatids are being pulled apart, pulled towards opposite ends of the cell by spindle fibers, another vocab word. Telophase, a new nuclear membrane starts to form around those chromosomes. And cytokinesis is when our new cells physically split in two. So that's a lot of words. If you need to go back and review the stages of mitosis, it's recommended. Don't spend too much time studying what happens at each specific stage though. For your 9th grade biology EOC, you want to focus on what happens, the overall end result of these types of cell division. So mitosis is going to allow for the growth of organisms. It is when we're replacing old or damaged cells. It's when organisms are growing. Meiosis, on the other hand, is when we are preparing to create new genetically different organisms and things like you and me. So we came from 2 different cells that were formed through meiosis. So egg cells and sperm cells are formed through meiosis, and that's because they are haploid. They have half the genetic information as the parent cell. Mitosis, like if my toe is just growing, that's going to be exact copies of the parent cell. But if you're creating those reproductive cells that are haploid, half the genetic information, it's going through meiosis. So my toe versus me for meiosis. Some people say meiosis. It's okay, it's up to you, but that's how I like to remember the difference. Now, again, you don't need to memorize all the stages of meiosis for your regular biology end of course exam. Maybe your teacher has you memorize them. Check with them. But one important thing you should remember that happens during the first round of divisions of meiosis is crossing over. So crossing over is when we're exchanging genetic information between homologous, the same type of chromosomes, and this is during meiosis, and this is going to allow for increased genetic variation. And this is part of why, our siblings don't look exactly the same. We're not just getting an exact same split of our mom's DNA and our dad's DNA every single time. We're getting a little mixed up version. So I might have a little bit from my grandmother and a little bit from my maternal grandfather. That mixing is happening in crossing over. So again, here's some vocab all together on screen. Meiosis, we're creating sperm and egg cells. Those will combine in fertilization to create a zygote, a fertilized egg. And then once that zygote is formed, it's going to start to do now mitosis, to divide and divide and divide with all the same combined DNA until it's time for those cells to differentiate. And that's when we're turning on different portions of that DNA to create different types of cells. So one cell might become a nerve cell, one cell might become a muscle cell, and eventually an entire multicellular diverse organism. All the DNA is still the same, just different parts of the DNA are being used in different parts of the body. Moving back a little bit to photosynthesis, you might just need to memorize this idea that it's not just food, it's glucose actually, that's what's being produced in photosynthesis. But we have sunlight and carbon dioxide and water, and the end result is glucose and oxygen. This takes place in the chloroplast, and what makes those green are the chlorophyll, which is a pigment that's going to absorb the sunlight in this process. Now, cellular respiration is kind of the inverse of photosynthesis, because this time we're taking glucose, we're taking oxygen, which we remember are the 2 products in photosynthesis, and they're being used to create water, carbon dioxide, and ATP energy. We always say how trees help us out by producing oxygen. They're going to use that oxygen as well because they also perform cellular respiration. Remember, plant cells have both mitochondria and chloroplasts. So they're doing both photosynthesis and cellular respiration. We just do cellular respiration. We have to get the glucose by eating it. Another type of respiration is anaerobic respiration, where no oxygen is needed. And so we might see this as fermentation, lactic acid fermentation, alcoholic fermentation, these different types of anaerobic processes. Now, this is when we don't use oxygen and less energy is generated out of these processes. So in general, you're only going to get 2 ATP per reaction from anaerobic respiration, where for aerobic cellular respiration, you're going to get a lot more, like 36, 38 ATP. All right, taking it way out large now to the macro scale, carbon cycle. So this is the movement of carbon through the environment. And photosynthesis and respiration are a part of this. So photosynthesis is going to take carbon dioxide out of the air. Respiration will release it. We are expelling carbon dioxide gas. In decomposition, that carbon might return to the earth, get turned into fossil fuels after many millions of years, and then it's burned and combustion occurs and that carbon goes back into the atmosphere. Sometimes dissolved carbon dioxide gets taken into the ocean and we have ocean acidification, but all of these things happening all the time across planet Earth. Another biogeochemical cycle, another big vocab word, is the nitrogen cycle. And while you don't need to memorize all the steps of the nitrogen cycle, it is important to know that nitrogen moves throughout the environment and we have bacteria that help us convert this nitrogen gas into something that is usable by plants, and then animals can eat it, go back into the soil. This also happens with lightning too. Lightning can help with splitting those nitrogen gas molecules apart. Now, this term ecosystem stability, that's the ability of an organism or of an ecosystem to resist or recover from disturbances. Usually, the more diverse an ecosystem is, the more resilient it is in the case of environmental change. All right, here's a couple different ecology-related phenomenon vocab you should probably know. Invasive species, these are a non-native species that are brought to an environment and cause harm. Biomagnification is when we have the increasing concentration of toxic substances at higher trophic levels because they're going to accumulate one level after another. Ocean acidification, that's when pH in the ocean is going to go up as the ocean absorbs excess carbon dioxide. Carbon dioxide in the water is going to turn into carbonic acid. A couple other chemical reactions are involved too. And then habitat loss. This is going to be our number one reason that an organism is likely to go extinct or become endangered. And so a lot of different reasons can cause habitat loss. All right, moving on to genetics. So you might have seen the words genotype, heterozygous, homozygous. These are just the combination of different versions of a gene or alleles that you can have for a particular trait. So for example, this is often represented as a letter. You can pause this and try to fill it in. Heterozygous means we have two different types of alleles. Homozygous, two of the same allele. So I'll fill this in here. You can pause it and check it out on your own. Phenotype is the actual physical characteristic that you will see based on a genotype. So for example, the genotype here, that's the letter combination. We're going to represent it by letters. And phenotype is the actual body color, the trait that comes from it. So in here, we will see each of these genotypes, big Y, big Y. The phenotype is the trait, yellow. These are all about color, but it can be about any specific trait within the body. Let's talk a little bit about Punnett squares. This is a tool to predict the potential combinations out of a particular cross. So if we have a trait like albinism, which is recessive, the regular pigmented axolotls here will have a dominant allele. And so that dominant allele, regular classic inheritance, like the first type you were probably taught, the dominant allele is going to mask the expression of that recessive allele. And so usually a big A, little a individual will have pigmented skin. The only option for getting an albino axolotl is to have two copies of that recessive allele. All right, I have other videos on genetics if you want to check those out. I don't want to get too deep in the weeds here because we have some more vocab to do. All right, codominance is when if we have two alleles, both of those alleles are going to be expressed. So here we would have a purple flower and a pink flower. If they were crossed, then in the offspring, we would see both the pink and the purple in the petals. Incomplete dominance is a blend. So that's when we would see a mixture of these two instead of one represented over the other or a combination of traits. Now, we also might see multiple alleles. That's when there would be more than just two different types of allelic combinations. So we'll see that in blood types where we have A, B, and O. Three different things leads to a lot of different blood type combinations. Environmental factors can influence gene expression. Gene expression is just how certain genes are turned on and off and how they show up within organisms. For example, this rabbit here in colder temperatures, they might have fur that turns white so that they can blend in and have better camouflage with the snow. There's a lot of different examples of this with like hydrangeas. If the soil pH is different, they can have different colored petals. And that's just how the environment is actually affecting phenotypes. It's not always just genes. All right, moving big again. Evolution is the change in genetic makeup of a population over time. Natural selection is how that happens. It's a major mechanism of evolution. Evolutionary fitness isn't how strong you are, how fast or big you are. It's measured by your reproductive success, how well organisms are able to survive and reproduce and have their offspring pass on their traits. There's lots of different types of evidence for evolution, from embryology, that's the study of embryos, biochemical evidence, geology, the fossil record. Fossils are traces or remains of ancient organisms that we can infer how they lived and how they've changed based on where they are in the rock and how they're situated. Vestigial structures are artifacts of common ancestry. So we might see a particular bone structure exists in one organism that we don't really use anymore, but it was there and useful to ancestors. Genetic drift, another vocab term you might have seen in the evolution area, is a change in allele frequencies in a population due to random chance. You might have talked about the bottleneck effect or the founder effect. It's going to have a bigger effect on smaller populations. This is not natural selection. It's just like a change in the genetic makeup of a population, usually because of that random chance. And then gene flow is usually just like about migration. So the movement of alleles between populations where individuals migrate and they're going to breed together. And this is just like one of the mechanisms of evolution that you probably studied. Sexual selection is where a type of natural selection where individuals with certain traits are more successful at attracting mates, so a certain color of peacock feather, for example. And then speciation is the process by which one species splits into two or more distinct species, often due to geographic isolation or natural selection. Cladogram is a diagram you might see like this. The lines don't represent time, but they are going to show us how evolutionary relationships are between different organisms based on their shared characteristics. And sometimes you might see little hash marks with traits marked on a cladogram as well. Binomial nomenclature, this is how we name organisms scientifically. So scientific names are assigned to organisms when we discover them. Usually this comes from their levels of classification at the genus and species level. So you might know that we are Homo sapiens because Homo is our genus, sapiens is our species. Technically, we're Homo sapiens sapiens. So for example, here Gallus gallus, that's the chicken. Sus scrofa, that's our boar here. Entamoeba histolytica is our amoeba, and Toxicodendron radicans is poison ivy. All of these organisms have their scientific names. Bi means two, binomial nomenclature. All right, biotechnology is the process of modifying organisms to create products or solutions for human use. A lot of times these are going to be medicines for healthcare, but it could be a lot of different techniques or applications to address lots of problems that humans have. And it's a growing, evolving field. There's a lot of techniques you might have studied when you talked about biotechnology, like gel electrophoresis, which you use in DNA fingerprinting. It's a technique that uses restriction enzymes to cut DNA into fragments, separated on that gel to create this banding pattern, and it helps us identify individuals, not just for crimes, but for evolutionary relationships or paternity testing. So there's a lot of different applications for that one biotechnology technique. You might have also heard of genetically modified organisms, and these are just an organism whose DNA has been altered using biotechnology, like pest-resistant crops or insulin-producing bacteria. You might have also talked about bioethics, which is the study of ethical issues that might come out of biotechnology. All right, and if you can believe it or not, we have gotten to the end of this vocabulary review. Give this video a like if it's been helpful, and I'll see you later.